Welcome back. Today we'll be going through a quick overview of metabolism. So, what's important to know is that metabolism is the totality of the chemical processes in the body, and each is unique to the individual. So, it doesn't mean that the way I metabolize, let's say glucose, is different from other people. We all metabolize the same, but the rates of which metabolism occurs is unique due to the way our genes express things. So, for males in particular, their metabolism is very focused towards anabolic reactions to build muscle, right? And that doesn't mean that women uh, don't. It doesn't mean that women don't catabolize things faster. We just don't have the capacity to build anabolically the way males do. It's important to know that there are some amphibolic uh, metabolism, which means that there are interconversions of the metabolites. This is fairly rare. And there are anaplerotic reactions, which replenish the main metabolism cycle, which is the TCA cycle. So, metabolites are, the results of each form of metabolism. The results, and by that I mean like the products and all of the transient products in between before we get to the final products. The metabolic cycle means that there's going to be the same first and final metabolite. So it's going to be consumed and recycled. The feeding metabolite are the metabolites that are consumed and don't they're not like replenished. We have to eat to replenish these metabolites. The anaplerotic metabolites are not consumed. These are the ones that undergo like isomerism. But the carbon skeleton itself is the same. It just goes through different transformations. Crossroad metabolites are the products that have different origins with several metabolic fates, and the examples are G6P, glucose, pyruvate, glycogen, and ribose 5 phosphate. So all of these are regulated by enzymes, of course, but in particularly the changes in enzyme concentration, the changes in enzyme activity, which results in inhibition. Hormones are another form of regulation. These are the long term um, controls, and nervous control is the short term control. So this is fast, this is slow. Let's go through carbohydrates really fast. So the metabolism really depends on the provision and the fate of glucose. So it depends on whether you are in a fasting state or whether you are in dire need of glucose, such as when you're about to go for a run. Glucose can be metabolized in pyruvate by glycolysis, and the pyruvate can go and become acetocoenzyme A, which goes into the TCA cycle, and then acetocoenzyme A itself can form like either fatty acids or they can actually make amino acids. And the glucose can turn back into lactate in glycolysis, and then the glucose can be transaminated to glycogen in the pentose phosphate pathway. So eventually we'll go through all of these, but what's important to know is that all of these are interconnected. So the body's way of responding to environmental stressors is quite remarkable. Just from a few things like glucose 6 phosphate, you can do so many things. Look at the arrows and go to like the pentose pathway, turn to pyruvate, which could go into anaerobic metabolism, or it can go into aerobic metabolism. So let's go into the lipids. Lipids are, of course, fatty acids, and they contain a whole lot of energy, much more than pyruvate or G6P. So the fatty acids are metabolized or oxidized, pardon me, to acetyl coenzyme A, and that goes straight into the citric acid cycle, or TCA. And from there, it can be esterified to triacylglycerol to make cholesterol, which could be stored either as fat cells, or they can be integrated into our cellular membrane. And from there, we can make ketone bodies or acetoacetate or 3-hydroxybutyrate in the liver. So there you go. So everything kind of starts with glucose, and then down, down, down. Acetyl A, as you can see. Citric acid cycle, and then in the end of all of this produces carbon dioxide. All right, so the process of which amino acids go through for metabolism is called transamination. So as you can see here, it can go into the acetyl coenzyme A, and then that in turn can go into the citric acid cycle. Or this is usually if you have consumed proper amount of amino acids and it needs to be excreted, so it gets deaminated into urea. So as you can see, amino acids are quite useful. So the liver is really important because it regulates the blood concentration of water-soluble metabolites. And by that, it controls essentially the catabolism or the anabolism of glycogen. It makes all the major plasma proteins, and it deaminates the amino acids in excess and makes urea. And the partial oxidation of fatty acids lead to ketogenesis. So yes, the ketone bodies come from the liver. The glycolysis pentose phosphate pathway, and fatty acid synthesis all occur in the cytosol of the hepatocytes, and those are managed by regulatory enzymes. And in the liver, this is the unique organ, because the metabolic fuels are interconvertible, and the exception is that once pyruvate has been committed into acetyl coenzyme A, it's reversible. So once the body has, oxidizes pyruvate into acetyl coenzyme A, that is it's kind of the point of no return. It's all or nothing. It's going to continue with the TCA cycle until there is some form of cellular signal that says, hey, stop, we have enough energy. And that will block the TCA cycle in one of the other regulatory steps, but it has already turned into coenzyme A. And coenzyme A can't be used for gluconeogenesis. And you can see here, lactate turned into that, amino acids, especially alanine. There you go, and then here you go. It turns into glucose 6-phosphate, glucose, then it is excreted. Or it can be stored as glycogen, which could be broken down from another, signaling cascade and then turned into glucose. So just to reiterate, most of the metabolic pathways are interconnected. So muscle. The muscle uses glucose aerobically to make a carbon dioxide. That's the final product. And anaerobically, of course, make lactate, and that's the cause of muscle soreness the next day. And the muscle stores glycogen as energy storage, and they make protein from the plasma amino acids. So they gather the plasma amino acids and kind of bring it into the muscle cell to catabolize. So here are the sources of ATP during exercise. And this is why people kind of focus on two different types of workouts. The HIIT training, so the high intensity interval training, and the long marathon kind of styles of training. HIIT workouts, of course, are very short, and this is the workouts that will, burn through previously made ATP. So this is like the stressor that makes you catabolize, or yes, catabolize the energy quite quickly. And then with the BCAAs and like creatine phosphate, that will provide energy for like a longer term muscle usage. And then past like hours, then it goes straight into like anaerobic metabolism. So the aerobic energy gives you quite a bit of energy, but it's only for a short amount of time. As you can see, it's just like seconds. And then you have to go into anaerobic metabolism, like not just like, in hours, but as you can see, it's just, minutes. You're already going into anaerobic metabolism. But as you can see here, it's not like one or the other. You do both. You have both like anaerobic and aerobic metabolism. The adipose tissues, are your fat tissues, and this is where the lipids are hydrolyzed, and then they are re-esterified in the intestines. So from there, it moves through the blood as chylomicrons, which will be cleared by the liver. So chylomicrons are just like tiny globules of lipids. And in the liver, they are metabolized by the lipoprotein lipase. Remember, lipase cleaves. Then the adipose triacylglycerol are hydrolyzed and transported by albumin to be esterified for storage or oxidized as fuel. So just know that it's once ingested, the enterocytes will re-esterify or hydrolyze and then re-esterify the lipids into chylomicrons, which will be transported essentially to the liver, and then from the liver it goes everywhere else, such as here to the brain, to the heart, to the muscle. Okay, insulin. Insulin, as everybody should know, is a indicator for blood glucose levels. So let's go through the mechanism of action. There's high amounts of glucose in the body, the GLUT4 transporter, it's an intracellular vesicle. It will help the glucose come into the cell, so outside, inside. So go into the cell where glucose can be utilized, or it is stored in as glycogen, or used in the synthesis of other things. But that is dependent on the insulin receptors. And when glucose levels are high, the vesicles, they're not always active, right? So they'll go forth from the inside of the cell into the to the surface, and they fuse with the membrane, and then those will expose the active glucose transporters. But that doesn't work until a signal comes and binds to it. So if a signal doesn't bind, they're going to be inactive. So once the insulin signal falls, there's no more glucose. The receptors will inactivate and then get internalized. So they'll be brought back into the cells. As you can know, insulin resistance can occur if there's too many of these inactive receptors on the surface of the cell, or in type 1, there's a genetic component where there aren't receptors produced at all. So in muscles, the calcium ions will signal the migration of the insulin receptors to the outside of the cell because of stressors. The uptake of glucose in the liver is independent of insulin, which is unique because it has glucokinase, which increases the glucose amount entering and synthesis of G6P for glycogenesis. So for the rest of the cells in the body, we have this kind of mechanism because we use glucose when we need it. But for the liver, that's kind of the storage system of the entire body. So glucose is constantly going in and out. That's why they have glucokinase instead, because we need a lot of glucose inside of the liver in order to either make glycogen or to produce other proteins, lipids. So in the liver, insulin will stimulate, or liver and muscle, insulin will stimulate glycogen synthase and inhibit glycogen phosphorylase. So glycogen synthase is to make it. So of course these have to be opposite. You can't make it and activate it at the same time. This has to be inactivated in order for glycogen to be made, because this will break down glycogen. In adipose tissues, insulin will stimulate glucose uptake, conversion to a fatty acid, and esterification to triacyl glycerol, and it will inhibit lipolysis. Of course, because the adipose tissue is the storage cells, and they will stimulate the glucose uptake to sequester it away. Adipose tissue and skeletal muscle fatty acids are used to taken up and used to make, once again, triacylglycerol, and then that is transported as LDL. So it's the low density lipoproteins, which is, of course, not good for you. Too much of the transport can build plaques on the sides of the arterial or the venous walls. So liver will take the glycerol part of the fatty acid for glycogen synthesis or lipogenesis, and then it will re-esterify the fatty acid, and the fatty acid can be metabolized in the body to make like ketones. So usually proteins don't have an increased rate of catabolism. It's fairly stable, unless you are purposely trying to bulk up. But proteins are generally just like, used and reused in a cycle. The concern is in cancer. So proteins actually, let me restate this. So proteins are in equilibrium in the body. Unless you are bulking up, then it will be an increased rate of anabolism. But normally, people do not have an increased rate of catabolism because we don't want our muscles to atrophy. This occurs only in like starvation or in cancer. So when you see like a long-term cancer patient, their muscles are atrophied, their skin is very frail, and it is called a cacaxis. So in the fasting state, the concentration of glucose in the portal blood coming from the small intestine falls, and then insulin secretion decreases. Glucagon instead will be secreted by the alpha cells of the pancreas. That inhibits glycogen synthetase and activates glycogen phosphorylase. So the opposite of what insulin does. Of course, because now we need energy. We are in an energy deficit. So the glucose 6-phosphate is hydrolyzed and then glucose is released. The muscle glycogen can't contribute directly to plasma glucose because it doesn't have the glucose 6-phosphatase. The muscle is quite selfish. It only kind of makes the protein or the amino acids and proteins for itself, and it only catabolizes those products for muscle use only. So the acetyl-coenzyme A in the muscle is transaminated to alanine, which is transaminated in the liver for gluconeogenesis. And a fall in insulin in the adipose tissue makes it release more glycerol and non-esterified fatty acids, which will be metabolized during glucose. This is kind of what us Americans are aiming for. We want to metabolize the fatty acids instead of glucose, and this will lead to like weight loss. So the liver is a nice guy. Liver can make more acetyl-coenzyme A than it can use, so it will make the ketone bodies during fasting, and that will go everywhere. So that would be used, in the brain, in the muscles, in the intestines, in order for the body to have energy. This doesn't occur until like almost like a week and a half of fasting. So the body doesn't actually make ketone bodies like in a significant amount until then. Here, once again, is another nice little chart. As you can see, so if this goes into the TCA cycle, it will be directly like, you know, the final product will be carbon dioxide. Everything will be oxidized. So these are the clinical aspects for metabolism problems. Death will occur when the essential tissue proteins are catabolized and not replaced. Prolonged starvation will catabolize the muscle tissue. And a constant high demand, I should say, for glucose will lead to ketosis, or, you know, diabetes 1. Metabolic diseases are caused by mutation, and they're almost always autosomal recessive. And those inborn errors will have like the phenotypical manifestations, which depend on like the substrate accumulation, the different kind of product deficiency, and the unique substrate properties. And that can affect the soluble small metabolites, and that will be a systemic problem. While macromolecules, if affected, it will only cause like localized symptoms. Enzymopathy is the disease process where there's a loss of enzyme activity, and that can be caused by either substrate accumulation or, you know, product deficiency. Phenotypic homology are when enzymes are working in the same area of metabolism, and it affects the similar steps but in different cycles. For example, dehydrogenases, those are used in several different pathways, but the mechanism of action is the same. You will dehydrate a molecule. That's it for now. Thank you for your attention and good luck studying.